Well, welcome back, everyone. We're going to go ahead and move on to our session 2, which is on computational approaches. And I wanted to introduce Dr. Andrea Baccarelli from Columbia University, and he will be the moderator for this session.
Thank you so much, Kim, for inviting me to be here. And it's my great pleasure to start us off with this session on computational approaches. We have 2 speakers who will have 20 minutes for their presentation, and then we have an amazing team of panelists who will guide us in our discussion. Without much ado, I would like to introduce Dr. James Gaderman, who's at the University of Southern California at the Keck School of Medicine, and is going to present statistical approaches for integrating environmental and omics data in cancer epidemiology studies. Thank you, Jim, for being with us, and I'll take it away.
All right, thank you, and many thanks to Leah and Kim for organizing such a great session. Yeah, so I'm going to give a sampling here of some various analytic approaches that we've thought about, especially related to how environment and genetics and omics perhaps can be used to advantage to try to understand risk factors, improve prognostic models, et cetera. I'll talk a little bit about data types, some selected approaches, and I'm going to focus in the areas of interactions and mediation, and then I'll end with a few opportunities. So we've all seen figures like this through the morning session, where we have this kind of amazing range of data types that are now able to be measured at various different levels of resolution. And with lots of data that can be produced. So when you start trying to put these things together, the mind starts to boggle and the computational resources get strained if you're going to try to look at all possible pairs or trios of various kinds of omics layers. So it really forces us to think about dimension reduction, feature selection, et cetera. I'm not going to talk a lot about those kinds of techniques. There's a very nice paper by Marilyn from a few years back that goes through and describes a lot of kind of interesting features and ways to kind of address multi-omic data. What I'm going to spend more time on is thinking about the environment here on the one side, the relationships to cancer, and then how we can think about bringing in various levels of omic data to try and inform the models and our understanding of what's going on. So being in Los Angeles, we spend a lot of time thinking about air pollution, and we've been doing air pollution research here for close to 30 years now, primarily related to respiratory outcomes, but now moving into other areas as well. So when you think about air pollution, it really is a good paradigm for the levels of data complexity and kind of measurement scales that you can have now for trying to get a sense for what a given individual might be exposed to. And so it ranges in the upper left here from fairly crude things like kids filling out questionnaires about smoking habits or housing characteristics to regional monitors that kind of give you a measurement, good time resolution, but spatial resolution, just kind of capturing general haze. Satellite measurements now getting a little more refined. We can geocode people, look at how close they live to freeways, maybe overlay some models for tailpipe emissions and try to personalize some effect estimates, or some exposure estimates. GIS and technologies allow us to track commuting patterns. We now have wearables, and then there's things like exhaled breath, NO, that we can use as biomarkers of exposure. So all of them come with certain characteristics in terms of more precision, but sometimes more measurement error. And as we talked a lot about this morning, the timing of measurements is going to be really important. When we think about omics data, I'm going to kind of use this graphic here to denote the kind of data we're collecting on samples of individuals from 1 to n, and then p different kinds of characteristics that might be measured within the omic profile. And that's kind of what the raw data looks like. We have the potential to then reduce dimension down to some layer of synthesis or layers. This round dot could indicate multiple layers. So things like clustering, principal components, sparse PCA, et cetera. Again, I'm not going to talk about these in any detail, but these are a wide range of techniques that can be used to try to reduce dimensions. Throughout my talk today, I'm going to bring in examples from a study that I work on with Ricky Peters and others, a big genetic epi study of colorectal cancer that is actually a consortium of, I think it's over 60 studies now, all contributing individual level data. So we have on over 120,000 subjects, genetic data, imputed SNPs, epi data that's been harmonized at the individual level on a whole host of colorectal cancer risk factors, and then some sub-studies of omics. So we have gene expression from a sub-study of normal colon tissue on 445 subjects. Included in that nicely is also the same exposures that we have in the broader epidemiology study. And then there's a colon organoid expression study on 34 pairs of colon organoids that Matt Duvall and Graham Casey have been working on. And you're going to probably hear more from Matt on this in tomorrow's talk. And the goals of this are to try to discover novel interactions, G by E or omic by E interactions. So I'm going to start really simple here. How does an exposure affect cancer? Well, this is kind of basic biostats here in epidemiology. There's case control, cohort studies, et cetera. E can be binary, continuous, categorical, and we typically use some kind of a fairly simple logistic linear Cox regression, depending on the nature of the outcome, to model the effect of E on the outcome. And we include covariates in there to adjust for confounders, et cetera, but I'm going to omit those for now. Turning to the Fiji study, this gives you a sense for the risk factors that I've shown you, straight E versus Y kinds of effects. And as you can see, all the kinds of things that we might expect for colorectal cancer BMI, that should be greater than 28 grams per day. Sorry about that. Heavy alcohol, smoking, male versus female, meat, and height are all on the risk factor side. And then there's a number of kind of factors that we all know, vegetables, fruit, moderate alcohol, et cetera, that are on the kind of the decreased risk side. As I mentioned, the goal of this study, though, is not these direct effects. It's to try to bring in other layers and understand interactions. So the one that we are all very familiar with is just standard GWAS. We have SNPs measured on everybody, as we do in this study here. Again, a simple GWAS is just looking at a kind of a logistic regression or linear regression or Cox regression, whatever it might be, and generating the kind of Manhattan plot that you see here, identifying any SNPs that show strong significance. So this is what the kind of the standard GWAS Manhattan plot looks like for colorectal cancer. But what we're really more interested in, I think, is how these factors kind of interact with one another, or how one might mediate the other. So I'll start kind of talking a bit about interactions here. And so, for example, the gene alcohol interaction work that we've done, there's really, as others have pointed out, the minute we start thinking about interactions, the power drops precipitously compared to looking at main effects. And we see this in a number of our scans for different exposures, where the Manhattan plot here on the right is for alcohol, for moderate alcohol. And it's not unlike what we see for several of the exposures. So it's got us thinking about analytic strategies to try and improve power somehow, just by thinking more creatively about the information within the data we've got that is informative. So we've come up with some two-step methods, others have as well. These are summarized in a review paper that a number of us participated in, that Kim and Leah spearheaded a workshop several years ago, and a number of papers came out on that topic, and other topics related to interactions. So the basic idea is to screen on SNPs that are most likely involved in interactions, rank them based on their step 1 p-value, and then look at significance in step 2 for an interaction test with differing significance levels that kind of take step 1 into account and use that information. So when we do that with the alcohol example, we go from seeing nothing statistically significant to seeing an interesting region in the COX-15 and ENTPD genes on chromosome 10. What about looking at direct effects of E on some omic layer? So this is a situation that probably Matt will be talking about, things like looking at organoids, pairing them off, exposing one member of each pair or one sample of each pair to a particular exposure, leaving the other unexposed, and then looking for differentially expressed genes. So now how about kind of adding 2 omics layers together? So the way we kind of think about it is, and a lot of others as well, are we thinking about integrating the layers kind of late in the analysis phase with respect to how we're looking at the outcome, or early? So examples of late integration would be kind of doing that G by E scan to start with SNPs, taking the top hits out, and then in a separate database, focusing just on maybe a handful of GWAS or GWIS significant SNPs, and looking to see whether any of them are interacting with E in terms of affecting expression. Or we could look at whether SNPs that show interaction in the main data, when you pull them out and look at those SNPs and flanking genes, develop EQTL models and corresponding PRS, bring those back to the main data, and do you see some expression by E interaction? So for example, I mentioned that the SNPs we found for the alcohol example in that COX-15 region, we did just that second approach. We pulled out some SNPs in that region, built some EQTL models using that separate study of 445 normal tissue samples, and then asked the question, do we see evidence for predicted expression at these loci that were indicated by the SNP scan be also interacting with alcohol? And we did. We saw the expected kind of moderate alcohol having a protective effect, and an additional protection that came as the expression in these loci increased. Okay, so that's kind of an example of late integration. What about early integration? So this is a situation where, you know, we might be looking at, say, single-cell RNA-seq and genome-wide SNPs, and looking at SNPs that show differential expression in one or more cell types that we might care about. And so, you know, one fairly simple approach would be to just identify those SNPs that are showing differential expression in some tissue or cell that we care about, focusing then our subsequent interaction analyses on just that subset. So it's a fairly draconian, you know, you're in the analysis or you're not in the analysis, but trying to bring in the upfront analysis with single cell data to try and help pare that list down. The other option would be to do a full TWIS. So try to build EQTL models genome-wide for all the genes, getting prediction models, which I'll denote by X hat, and then doing kind of a transcriptome-wide interaction study. We could think about, so in this latter approach here, I was thinking only about EQTLs based on the standard kind of G2 expression models that we would tend to build, looking at probably cis EQTLs. We could also think about looping exposure data into that QTL model building exercise. So in the example I mentioned, where we have 445 normal colon cells, normal colon samples, sorry, and we have the exposure data there, we could think about augmenting our EQTL model to bring exposure in at that stage and try to build perhaps a better polygenic risk score based prediction model for expression, and then go forward and do a kind of a TWIS. And then there's the potential, rather than doing it in stages, to try and think of it as one combined analysis. So rather than do step one, taking something forward, trying to put it all in one big model. And so we've been doing a little work here using Lasso and extensions to Lasso to build a model that has a function of interactions and main effects, incorporates main effect before interaction kind of hierarchy constraints, and then integrates external gene expression to guide the selection of interactions. That becomes a computationally challenging problem. So we're not talking about being able to analyze millions of SNPs and tens of thousands of expression profiles simultaneously. The data has to be cut down in some way in advance. What about exposures? Well, I've been thinking so far about exposures as just single variables, but we may want to think about those as maybe flawed indicators of some underlying thing like lifestyle that might be more important. So this is just one example of something we could do. We could take the exposures we have in this Fiji study, run a standard principal components analysis on them, and try to pull out some common features that maybe could be carried forward and then used in downstream interaction analysis. So, it has the vagaries that principal components analysis always does, that you want to try to put labels on these things and try and understand what a particular principal component might be telling you. But if you see something down the line, you can always go back and try and tease things apart and try to understand it a little more fully. Another potential is to try to integrate exposures by computing an exposure risk score. So similar to what we do with polygenic risk scores, we fit a model with all the exposure variables up front, take the linear predictor out of that, and then apply that to create a score for every individual back into the original sample. So this is what, for example, a distribution of an environmental risk score across those 12 or 13 exposures that I've been talking about looks like in the Fiji sample. And you could then take that forward and try to look at interactions with either SNPs or with some omic layer. And then what about the situations where we want to look at, say, a biomarker, or we have incomplete E data or maybe no E data in the main sample, but we do have it in some external resource. So we can think about potentially building an exposure model. So for example, people have looked at looking at GWAS SNPs for things like CRP or glucose or IGF-1, et cetera. Models have been built using UK Biobank, and we can think then about applying those models back into our sample to generate predicted biomarker values for everybody in our data set. So for example, we're looking right now, doing some analysis of IGF-1 using a prediction model that came out of this paper referenced down here, and then trying to take that forward and look at whether we see interactions between SNPs and expression with that kind of predicted IGF going forward. Notice though that we lose something, obviously, because we know up front from cross-classification analysis that the R-squared in the original source population used to build the model, that they're not that good for some of the kind of biomarkers that we might want to follow up. So we need to kind of keep that in mind. I'll talk real quickly here about mediation. So here the goal is to try and see whether exposure is mediated through one or more layers of some omics, contrasting and comparing the direct effect with kind of the intermediate effects that we can estimate. There's a nice paper by Max Ong and some of his colleagues that look at several exposome values and biomarker values And they come up with kind of a really nice way to think about going through this, either looking at all possible pairs of 38 exposure analytes and 61 biomarkers, or doing some upfront dimension reduction on the exposure side to come up with 4 kind of layers here, and then doing the 4 times 61 analysis, or doing dimension reduction also on both layers, and then narrowing it down to only 24 analyses. So they have some nice results there, and a nice discussion about what's gained and what's lost with each of those layers. And then again, that's kind of a, doing it in stages, step 1, step 2, dimension reduction, and going forward. We've been doing work here about trying to integrate it into one single model, an approach called Lucid, where we're trying to put it all into one kind of analysis and get nice pictures like this, where we can have some exposure, some biomarkers that are measured, some outcome that's measured, and then try to estimate unknown clusters that help link the exposure to both the biomarkers and to the outcome. And then there's thinking about PRS. So I probably won't have too much time to talk about this. It looks like I'm out of time. So maybe I'll stop here and turn it back to Andrea to go to the next section.
Thank you so much, Jim. That was so clear and amazing, and I learned a lot, and I hope everyone did. And there are a couple of questions. We have 10 minutes for questions, so any questions you might have, write them in the chat. The first question is about your the 2 steps approach that you showed for GE interactions. Are we assuming all interactions happen to only for those significant SNPs in step 1? And you show different scenarios. This may be an earlier question.
Yeah, that's a really good question. If I can just go back really quickly, the answer is no. What happens is you take all of the SNPs out of step 1, you do something like a GWAS, so that gives you their GWAS p-values, and you use those then to rank order them into bins. So every SNP gets analyzed in step 2. This is showing their G by E p-value in step 2, and the payoff from using step 1 is that you can set different thresholds of significance. So those SNPs that are in the upper bins, meaning they were more significant in step 1, are being evaluated in step 2 against a lighter significance threshold. Maintaining, and the important part of these is that you maintain the overall type 1 error rate to be 5%.
Thank you. That was really helpful. Another question is, I was wondering if you could comment on the tension between dimensional reduction versus the need to know what is actually captured in those scores to inform mechanistic hypotheses. How have you chosen where to reduce dimensionality versus where to preserve some complexity in your GE work?
That's really an important question. It's really it's hard to answer that in general, because I think it really is context specific, and it really depends on the type of data that you've got. I think there are some situations where keeping things at the lowest possible level you can may work, but again, you get to some of these situations where you have millions of SNPs and tens of thousands of genes and maybe some other biomarkers, and it's just not possible to do that. So I think part of the art here is to try to figure out ways at each of those layers to pull out the relevant information and then to carry that forward. How to do that, I don't think there's a good general way to give advice. It really just, I think, depends on the particular application, and it does really put a premium, though, I think, on taking time with the analysis and not just kind of rushing to the final. I want to get the outcome, and I want to look at the exposure, and I want to get an answer fairly quickly. I think it really puts a premium on understanding each of the analyses, the steps that might go into that, and try to carefully understand what might be lost or what might be gained by dimension reduction.
Fantastic. Thank you so much. I really appreciate also the point about taking your time in thinking about your analysis. There is a question from Jay Lubin. One additional issue that is generally ignored is the form of association of an individual exposure with outcome, and the impact that any misspecification of the form can have on the G/E assessment. In my experience, a RR association is seldom, if ever, log linear over the full range of a continuous exposure. Examples are radiation and smoking with cancer outcomes. The use of incorrect modeling of exposure risk can itself distort results. So it's a comment for your consideration. I don't know whether you have any further thoughts on this.
That's a great comment, Jay. And I know the work you've done, the great work you've done in trying to understand those kinds of relationships. And I really agree with the idea that I think, we have to be careful when we get to these large scale omics studies that we don't lose sight of, this kind of thing where the form of the model really is important. And we tend to, kind of boil these things down to a simple logistic model, binary exposure or a linear trend or something like this. And we do that partially for expediency because we're trying to deal with the magnitude and the computational issues and things like this. But it kind of gets back to what I was saying earlier, that I think real care needs to be taken at each of these steps of the analysis. And this is a really good thing to think about, is exactly that relationship between exposure and whatever outcome you might be measuring, trying to get that right.
Thank you. I would like to add here from the Zoom Insider team, there is a request of whether you could actually comment on the PRS, if there is time, which we have.
I'd love to actually. Yeah, if you can indulge me for just a second.
Absolutely.
Okay, I'll try and get to that. All right, so basic PRS. So GWAS SNPs, you form a linear predictor out of the significant SNPs or flanking SNPs or some large collection. You end up with this kind of an expression. And people are starting to use these not only to look at direct risk on some outcome, but to also think about how to use those for interactions or for effect mediation. And the problems that we see, if you just go back to the original kind of how you lay out the model here, what you're boiling perhaps tens or hundreds of SNPs down to one measure. And so when you are estimating the main effect, the interaction, you're assuming that you have the same main and interaction effect across all of the SNPs that are in that PRS. But it's very likely, we talked a little bit about that this morning, that the SNPs that form a PRS include SNPs with very different pathways. So one solution that we see is to kind of think about breaking the PRS up into subgroups. So subgroup A, subgroup B, et cetera. And rather than modeling a single PRS, thinking about multiple PRS and multiple terms in a model, which still is going to have a much lower dimension than putting hundreds of SNPs in, for example. And then the challenge becomes, how do you figure out the subsets? So is it SNPs that are also linked to related traits or EQTLs or common functional annotation? So we did this PRS by BMI interaction. It's kind of a first use case example here. In the Fiji study, saw absolutely no evidence of an interaction between PRS and BMI, p-value 0.91. And so I worked with my colleague here, Yu Mi, and his group, who have some nice tools for annotating. They took the 203 GWAS significant SNPs that are now known, annotated 153 of them. They were able to annotate to genes, and then did a Panther analysis and identified 4 pathways that were represented or overrepresented by the GWAS SNPs. So I looked at one, the TGF-beta signaling pathway, and it included 8 of the 200 SNPs. I looked at a PRS just based on those 8 SNPs, came back and looked at the BMI interaction and see some significant evidence. So very preliminary analysis, but a suggestion that if we think a little bit more carefully about how we construct PRS and what it's going to be used for going forward in terms of interactions, we may gain some clarity and some precision on what we're actually measuring through a PRS that could have some positive effects down the line.
Thank you. I'm glad we got to see that. Very interesting. And we still have 1 minute, I think. If there are no other questions, perhaps I can ask one, which is, we have a lot of people on Zoom or the webinar in our labs who are eager to learn these methods. How do we train, especially younger people who want to learn to do this? How do you do that in your lab?
Well, I mean, we have a PhD program, so we teach a lot of the stuff in our PhD program. So you can send them to USC. There are workshops. We participate in a workshop through Columbia and the SHARP program. In fact, we just did a 3-day workshop a couple of weeks back, and we're going to do another one in June, where we take 3 days, and several of us that work in these areas kind of step through interactions and mediation and PRS and population kinds of level analysis, and pair that with some labs where we teach students how to use software to carry out some of these analyses. So, you know, there's that kind of opportunity for folks that have some statistical savvy. We assume a little bit, but then we try to step through techniques.
Fantastic. Thank you so much, Jim. This was wonderful. And I unfortunately to wrap up here, but we have another exciting presentation coming up. And I want to call up Dr. Christian Coarfa, who is at Baylor College of Medicine, and he's going to present his NIHS funded research on longitudinal multi-omic characterization of a community cohort after chemical exposures from Hurricane Harvey. And we look forward to your presentation, Christian.
Hi, thank you, Andrea, for the kind introduction, and I want to thank the organizers for the chance to present our work. Without further ado, so in our case, what prompted this was a natural disaster. So Hurricane Harvey hit Houston in August 2017, dropped over 50 inches of rain, causing catastrophic flooding. Among others, it flooded over 100,000 homes in Harris County alone. This is the county that encompasses Houston. And then during this flooding, there were a number of other occurrences of public health concerns. Particularly, there was floodwater contamination, almost a toxic soup. Again, Houston as a city houses a lot of petrochemical industries, so there was a lot of contamination. Don't forget, Houston has a number of Superfund sites. So, and also flooding led to mold and mildew exposure. And after this disaster, a group was put together, led by a number of people at Baylor, Dr. Walker, Dr. Bondi, Dr. Simansky, to try to learn from the outcomes and then learn from the environmental exposure of people who are affected. Such a study was set up called Houston Hurricane Harvey Health, or Houston 3H. By community engagement, this group was able to enroll a number of people from Harvey exposed households. And then as far as the recruitment was about 200 participants who were measured with different omics about 1 month after Harvey, and we call this time 1. And then some of them were followed up. So about 125 were followed up in both time points. 141 new participants were enrolled. And the information compiled was multidimensional. So one was based on questionnaire and geographic information system describing the flooding experience. Another one was health outcomes. Now, this was fairly limited, focused on allergic symptoms, injury, mental health. No electronic medical records were being checked. As far as biospecimens, a large focus was on fungal microbiome and bacterial microbiome, focus on household swabs, nasal, saliva, and stool. And something which was fairly innovative was the use of a wristband sampler. which essentially sequesters chemicals, and then after mass spec analysis can detect over an inventory of over 1,000 chemicals. And then the areas where what people call concentrated had different properties, and this from the analysis. So one of them was the Addicts Reservoir would lead to flooding because the reservoir had to be released to prevent, catastrophic damage. This was an area with middle income. Bel Air, Maryland area, this is a fairly affluent area. A lot of the people who live there were medical center professionals. East Houston, this is a low economically depressed area, predominantly minority. And Baytown, which is really close to two Superfund sites. And there are a few others area which are not in these 4 main locations. And in terms of the data collected, just to review for the audience who might be less familiar. So we looked at microbiome using a 16S ribosomal RNA sequencing, which is a fairly low resolution profiling, achieving genus level resolution. We looked at ITS, internal transcript spacer sequencing, which is similar to 16S with a focus on regions variable in the microbiome and fungi. And we have done also fairly high resolution whole genome sequencing of the microbiome. And the data was summarized in 2 ways, using at the species level, using Metaflan, and at pathway level, using human. And this is just a presentation. So when you put this data, we appreciated that we are able to assess and quantify both the intrinsic environment, including demographics like race and ethnicity. And you can see the nasal, saliva, and fecal microbiome and microbiome, as well as the extrinsic environment, which includes socioeconomic status, diet, health history, and includes the home environment measured using 16S bacterial and ITS2 fungal assays, as well as risk band for chemical exposure. Again, this was a study performed with volunteer participants, and as such, we had less control over the completeness of the data. And you can see that in terms of samples, certain types of omics like environment or fecal had more samples, other had fewer. And also at time 2, we are able to get significantly more participants than at time 1. And remember that at time 1, some of these people who are flooded, they were trying to contribute, but at the same time, they were dealing with major disruptions in their lives, okay? In terms of complexity of the data obtained, you can see that maybe not surprisingly, environmental assays like 16S and ITS gave us the highest number of features. Actually, same with the nasal, and nasal stays at the gateway between the host and the environment. So that was actually reassuring that we are getting the type of results that we are expecting. And then very important in the process as outcome variables, we looked at questionnaire data. So this questionnaire data spanned a number of different aspects. So one was demographics, area deprivation index, age, diet. Very important was the Harvey exposure. We get metrics of pollution burden. Some of them were computed, some of them were measured. And as far as the health outcomes, there was a fairly reduced set, again, mostly focused on stress, injury, no use of medical records, electronic medical records. So with that in mind, we're going to present our types of analysis, and we're going to build it up from simple to complex. Now, it doesn't mean that any of them is not informative. We found all of them quite informative, but for the purpose of this meeting, we're going to show how we can build up such analysis. So first of all, we wanted to look at simply the correlation between the traits independent of what we measure in those individuals. And this is an important point because as humans, we might be aware of a couple of correlations. As humans, we are not thinking about end-to-end correlation, okay? So when you look at the correlation of the questionnaire traits, we group them again under diet, demographics, Harvey exposure, health, pollution burden. And we saw quite a number of associations, some positive, some negative. These were filtered for multiple hypothesis testing correction at an FDR of 5%. And with such an approach, then we can tease apart different aspects and dig deeper into them. So for example, one aspect was that of race and ethnicity. And we noticed that if we look at the participants which are African-American, we saw a lot of correlation with chemical exposure for that associate with the trait of, the ethnic trait. On the other hand, we also looked at correlations of with particulate matter and then with ToxConc, which is toxic water discharge. And we see, for example, that exposure to particulate matter correlates very strongly with participants in East Houston flooded. So again, these are very interesting aspects, can be delved and dissected further. And I think trying to understand formally the metadata is something that in most cases people just glaze through and say, okay, I can find an association on this variable, let me write about it. But I think it's important to also look thoughtfully and carefully into how the different metadata features talk to each other. All right, so going forward, we'll profile a lot of microbiome and microbiome data, and one of the very basic metrics is that of alpha diversity. Definition is that alpha diversity is the mean species diversity in a site of local scale. And interestingly enough, alpha diversity as a concept doesn't have to be applied only to microbiome, can be also applied to chemical wristband data. So we used alpha diversity in two ways. We wanted to understand how it relates to flooding and how it relates to neighborhood. So looking at flooded, we noticed that actually the fungal environment associated significantly at time one of flooding, we have a decrease in diversity. We saw a lot of changes in diversity from time one to time two. And interestingly, when you look at chemicals, we see significant, small scale, but very significant changes in chemical diversity between time one and 12 months later. Similarly, when you look at the neighborhood, we saw that, again, maybe not surprisingly, the environmental metrics and the nasal metrics correlate strongly with differences between neighborhoods at time one, right after Harvey. And a metric like ITS, the fungal environment, captures differences across the different time points for almost every neighborhood. So again, this speaks to the power of this multi-omic microbiome-focused approach. All right. So after alpha diversity, again, going to simple to complex, the next metric was basic differential expression. For this one, we used the 2A ANOVA, and we looked at two factors: neighborhood. Again, we have 4 neighborhoods: Addicts, Bel Air, Baytown, East Houston, and others. Flooding, yes or no. And we looked at also the interaction between those two. And when you look at the number of features, and remember that we measure, almost thousands of features in the environmental sample. So not surprisingly, we have a lot of differential expression within the environmental samples. However, if we look at the data, as a percentage of features measured, then we see that overall, the differences are not quite as dramatic. So we get anywhere between 5 to 10% of the features showing up as significant. And then trying to dig deeper, I'm going to focus on differential expression within chemicals. So we looked at the differences between neighborhoods, and this is, fascinating because we see, for example, that there's a number of chemicals, heat map has a representation. There's a number of chemicals which are common between the two areas, Baytown and East Houston, which are characterized by lower socioeconomic status. On the other hand, we also see some features which are specific to one area. And then we also look at took another perspective. So we try to say, are there features that are associated to neighborhood which are specific to only the time one, immediately after flooding, versus those that can be detected almost 1 year later? So doing a simple Venn diagram, we first identify the chemicals which are neighborhood associated at time one, so mostly flooding, versus those that are associated at time two, which might persist after flooding or might be not flooding associated. So we got this list, and then technically working with the developers of the wristband technology, Kim Anderson from OSU, we are able to try to identify which categories of chemicals these chemicals belong to. So interestingly, we saw that chemicals that are associated to neighborhood right after flooding, they span a number of different categories, but in particular, we are interested in polycyclic aromatic hydrocarbons and industrial compounds, which are known pollution risk factors in Houston. Right. So moving further, yet another, in terms of complexity increase, yet another approach was network-based analysis. We used WGCNA, which stands for Weighted Correlation Network Analysis. And the insight here is that in many systems, there's different features correlate strongly with each other. And then trying to first distill a system at a cluster of groups of features that correlate might allow us to get easier insight into what those features are doing. So what we've done first, we try to, for every omic, we try to identify correlated features. And WGCNA and related methods, they really like to use color names. So, you read papers talking about the blue module and the red module and the yellow module. So this is how this method works. On the other hand, the power comes that once we identify these modules, then we try to do numerical association using Spearman correlation with numerical traits. And then we get significant associations. Some are positive, some are negative, and we try to interpret them. So then the first thing that we've done is, again, look at all the data. I really like the previous speaker that was in a very thoughtful comment. You always need to take your time and look at the data to see what you're seeing. So in this case, when you look at the different omics, including the chemical, we notice that for certain types of features, there's a lot of features that don't correlate to each other in the chemical, in the fecal 16S. On the other hand, if you look at fungi, if you look at the nasal samples, or if you look at the gut pathways, almost everything, correlates to something else. So after that, we wanted to see what are the correlation between modules and clinical traits. And with correlation, another concern is that we don't want to identify weak correlation. So we put an additional filter that the goodness of fit of the correlation, the R-square, should be at least 5%. Using and we use the Spearman correlation because this is such a diverse data, we don't want to make any assumptions. And interestingly, some of the strongest associations by R-square are actually with the East Houston, which seem to be a site of particular challenges. And in addition to East Houston, we got associations in the fungal nasal, 16S saliva. Another area where we got some strong associations were the area deprivation index. We can dig a bit deeper into some of them. Again, WGCNA allows you to get some exploration and identify the features in a module, and then look at how individual features correlate to the outcome of interest. So in this case, we looked at the correlation between features from a yellow module with East Houston. In addition to the top features, we are particularly interested in associated with particular metal and toxic water discharge. So with that in mind, we looked at the association of toxic water discharge, and interestingly, in the blue module, some of the top features, Vicinami bacterialis and Acidobacteriota, are both soil bacteria. So this can be explained that, toxic water discharge brought some mud, and that, contain these bacteria, which are normally found in soil. Similarly, we looked at the association of particular matter. Interestingly, one of the top hits was fungus curvularia, which is present in mold. So, high level, we found some interesting association in an exploratory manner. However, also important, most of these associations have fairly low R-square in the generously around the 10%. So this low R-square motivated us to then use more powerful methods like machine learning. And then again, I'll give a very brief overview of our approach. I'm sure the computational side of the audience is well familiar with this. So we focused on 3 methods: random forest, which uses decision trees; support vector machines, which is essentially a hyperplane-based linear algebra modeling; k-nearest neighbor, which is a voting scheme. So with these 3 methods, we're able to do both classification for binary variables and regression for numerical variables. We didn't have a validation cohort, so instead we use a very stringent cross-validation. For 100 iterations, we split the data into 70% training and then 30% testing. And then depending on the approach, the metrics were area under the curve for the classification and R-squared for regression. And we'll start with the regression results. When you look at, we try to highlight here a strong association. So just looking high level, we see that the chemicals give a lot of interesting strong associations. The fungal nasal give associations. And actually, if you look at the features with a lot of significant associations across the board, is aging years, okay? So again, association between microbiota and age was reassuring. This has been reported before, and age is a powerful factor, leads to changes in DNA methylation. There's all kinds of clocks. But for the purpose of this talk, we are interested in association between particulate matter and then chemical exposure. So when you look at them, we see that the second most informative feature is benzotiazole, which is an industrial compound, indicating, not surprisingly, that exposure to industrial compounds can lead to particulate matter pollution. The other part of this analysis, we use machine learning in a classification setting, where again, we try to use the area under the curve AUC as a metric of goodness of result. And then what we notice high level is that in association with demographics, the chemical wristband and the nasal fungal were actually lead to strong associations. And actually, these associations I found interesting were much stronger than the ones that we determine simply by measuring the environment, which points that actually these demographics, they have profound impacts, and they lead to profound changes in individuals across its entire lifespan, not just where somebody lives, but where somebody works, where somebody travels, where they do their shopping, etc., etc. Okay, so this is actually fascinating. And then digging deeper, I'm going to show the results for association with area deprivation index. Again, I mentioned that the East Houston community is economically depressed. So not surprisingly, some of the top features that allow us to understand the area deprivation index group show up also when you try to understand the East Houston area. So there's a commonality. However, when you look at chemicals, again, we see that some of the top hits are the same. So Lilial and tonalide, and I looked it up. So Lilial is used in fragrances. Tonalide is usually used in laundry to provide some fragrance. However, when you look at the chemicals, we notice that East Houston also has a highly informative chemical, is pyrene, which is a pH. So this is saying that it's very important to do a multidimensional profiling, because the microbiome exposure would not capture the exposure to particular chemicals like pH. So with this, I'll try to summarize, and Lisa, go to the question section. So essentially, Houston was exposed to a natural disaster, which led to high exposure in a reduced time frame. Through community engagement and questionnaire-based data, we obtained a lot of information from our participants. The profiling of Omics was robust, focused on microbiome, and chemical wristbands. The methodologies used here range from simple to complex, so starting from alpha diversity and differential expression to network analysis and machine learning. And we use extensive cross-validation. And in terms of lessons going forward, we did not have the same omics in different omics in the same individuals. This is something we'd like to have in the future. I mean, we might use some mitigation to single cell approaches that match multiple modalities. And obviously, I think, we don't explain all the variations. So definitely we need to have more omics, like proteomics in urine, serum, blood RNA, cell-free RNA. And I want to say that this couldn't have been done without the generous help from NIEHS, which provided multiple generous grant support with the P30 Center grant, multiple disaster rapid response R21s. And I'm highlighting here the leaders of the various teams involved in collecting this data and putting it together. And with this, I think I'm ready to take questions.
Thank you so much, Christian. That was a masterful presentation with a lot of data. Congratulations on all the work that you and your team did on this. And we have some minutes for questions, and would encourage everyone to add their questions in the Q&A box. Meanwhile, I would like to start from a question that came up earlier that I think applies also to your data and certainly applies to your expertise, which is what would be the implications of the second human genome, meaning the microbiome metagenome, for the study of omics in general and GBA specifically for assessing predicting cancer risk? Are there good omics markers for the human microbiome?
I mean, that's a great question. On the other hand, it's a very open question. So we have been personally involved in microbiome-based data science over the past 3-4 years, looking for biomarkers associated with a wide range of features, vaccine response to psychiatric treatment, and what have you. And then the couple of thoughts, I can't answer this question, but I can give you some of my observations. So one is that microbiome is an exciting data, but it's also very noisy. So one of the challenges, if you want to rely on the microbiome, then you really need the power of many samples. This can be achieved in 2 ways. One, making sure you have many participants, but the other one is making sure you have longitudinal sampling, which might allow you, because even within the same individual, we notice that from time point to time point, there is fairly large variability in the microbiome. So many samples, many donors, many time points, this might allow you to at least, overcome the inherent noisiness of the data. Second, so then, we are able to find some biomarkers that, pass the eye test to consider distribution. And I think what is fascinating is that, and we're trying to do this in different systems, we're just trying to think to implement interventions. So whenever feasible, a probiotic intervention where you try to mimic a culture in somebody's gut is potentially a way to buffer against risk. Another approach is actually, again, certain communities are, say, immunocompromised, would not appreciate being given probiotics. So then the approach is actually to try to identify what is the function of a microbiome or a collection of species and try to give metabolites or precursors that mimic that. And then, again, I'm a computer scientist, but I'm learning all the time from my colleagues. There's approaches like phages, which actually work in the opposite direction, trying to suppress activity of certain species. So in terms of biomarkers, in terms of interventions, I think there's a lot of potential to mitigate risk, to prevent, but nothing comes for free. So you need lots of samples and, or at least, donors and longitudinal time points. Yeah.
Thank you so much, Christian, for that. There is a question in the chat by Ronnie Zhang about whether are the serum or blood metabolites incorporated in the network analysis or integration analysis or machine learning as well.
But we did not compile, blood or serum metabolites. We are involved in other work in other systems like prostate cancer or liver cancer, where we're actually using those. So the answer is yes, definitely, when such data is available, we definitely want to incorporate it. And I think I kind of alluded to one thing that one real challenge, whenever you work with volunteers that might or might not participate at every time point or might skip something, is that actually we don't have, often we don't have a comprehensive data. So TCGA was able to obtain the cancer genome models, obtain tumors, and they run in the same tumor biopsies, RNA-seq, DNA methylation, microRNA, what have you. So then we can really do multi-omic correlates in the same individuals. I think, and I kind of alluded, I think the closest thing I'm seeing at the time being that, might provide, methodology is actually the single cell approaches where people are trying to combine site-seq with single RNA, with spatial, with others. So I think some of those methodologies can be applied, for example, in dealing with sparse data where you don't have all the omics in all the subjects. I think actually this is something I'm interested in. Again, it's interesting how our field provides many opportunities for cross-pollination, intellectually speaking, Yeah.
Thank you so much. And there is also a question here from the control room, which is, we all envy you and your colleagues because we have a lot of data and very detailed characterization in this study. It's a unique study, but is there any knowledge that can be brought down to studies that are not that lucky and don't have detailed characterization of the exposures or the phenotypes? And what would you say to people who might have data sets that are less detailed?
I mean, it depends on when you plan. So I saw, for example, that if you have to prioritize for this environmental exposure, it really seemed that the most informative sources were the chemical wristbands and the nasal microbiome. So I think if you want to prioritize, if you something like this, those are, you might want to start with the features where you get more, more information and, so more bang for your buck, if you will. The other one was that, I mean, again, I think that it's sometimes it might be easier to obtain the detailed trait information, okay? So I would say that if anything, try to focus your efforts into samples where you have access to a lot of clinical information. Because in that sense, again, from a self-serving perspective, okay, there's a higher chance to find some interesting associations, but realistically, from a scientific, very personal perspective, I think, understanding really well your participants is actually crucial, okay? So like fewer omics, but detailed, metadata, that to me is actually very important, yeah.
And perhaps even from the other way around, I mean, we know that we spend months and years often designing population studies and thinking about all the different data we can collect. But then when you do data analysis and data go to teams like yours, that is when really things start to unfold and you might realize that there are data that were important that were not collected 5 or 10 or 15 years ago. Is there anything you wish you would have collected in this data? And if you had suggestions for people starting new studies, what are the data we all should collect?
I mean, first of all, a team like mine doesn't include only my team. Like I am meeting regularly with a number of these, people who I show, Dr. Walker, Dr. Simansky, Dr. Petrosyan, and others, so we always bounce ideas. I mean, us, based on their availability, but so there's a constant feedback, I meet them at least, once every other month. Some of them I meet like, on a weekly basis. So definitely these are, try to put together a team where people talk to each other. It's an important part of the process. I mean, which is not really data, it's more logistics. But in terms of data, I showed that we don't have 100% AUCs. You only have like 20 to 30% R-squared. So definitely there's room for improvement. And I would say, frankly, it's trial and error. So like, if you ask me, I mean, I would like, for example, since you and I share an interest in epigenomics, I would like to do methylation with metastable epialus, I would like to do that, but because, I think that would capture inter-individual variability and would be informative. But practically, again, we don't know where the information will be. So if I will at least try to explore the serum metabolomics. I think this is actually particularly important and captures a lot of the, host. And then I would like to look at the blood epigenomics. Yeah, so if I would pick two, I would pick those. But by no means, I mean, I'm speculating they might be helpful in our system, but also it's trial and error, and we all learn from everybody's work, Yeah.
Thank you for saying that and say epigenomics is important. I hope you didn't do it because I'm here, but thank you for that. And
No, Andrea, I'm doing mostly because my colleague Dr. Walker is on the call. Okay.
Okay, we all make Cheryl happy. This
Yes.
is what we do for a living. And thank you so much. I think we are closing here to the end of the session. I'll pass it to Leah at this point. Thank you so much, Christian, for presenting.
Great, thanks for great presentations. We have a short stretch break. We'll be back again at 2:50 for the panel discussion. So please come back in about 15 minutes.
We are ready. We had two amazing presentations to kick us off with the part about the computation, and now we have a great panel, and I look forward to a very alive and useful discussion. I would like to briefly just introduce our panelists, who include Marvin Ritchie from the University of Pennsylvania, the School of Medicine, Nilanja Chatterjee from Johns Hopkins University at the Bloomberg School of Public Health, and Marina Sirota from University of California, San Francisco at the Becker Computational Health Sciences Institute, and finally, Anshul Kundachi from Stanford. So I want to get us started, and I'll provoke everyone with a first question to get the conversation going. And this is going to be about some of the analytical challenges that we have in trying to integrate omics data, which are typically collected on limited sample sizes while we are working on much larger epidemiological studies. And so I would like to hear from some of you or all of you, and any thoughts you might want to share about this is particularly welcome. Marlene, you want to go first?
Sure, I would be happy to go first. So I've been thinking about this topic for quite a long time, and I think one of the big challenges that we face is actually capturing the data so that we can then integrate it into our analyses. And one of the places where I do a lot of the kind of data analysis projects that I work in is leveraging phenotypic information from electronic health records. And electronic health records are really great for capturing clinical information like diagnoses, clinical lab measurements, procedures, and things like that. But they're not necessarily as rich in terms of environmental data. And when I say environment, I'm thinking not only some of the exposures that we heard about earlier, air pollutants and water pollutants, but also our social environment, social determinants of health, as well as social and behavioral factors. But what I've been thinking a lot about is how we can leverage the data that we do have to try to capture some of those other variables. And so a couple of thoughts. One is, and Jim mentioned this really briefly in his presentation about looking at geocoding. So the location of where people live is actually something that we do capture, not only in most of our research studies, if it's an epidemiologic cohort, but certainly in electronic health records, we have participants or patients' addresses. We can use those locations and use geocoding technologies to map them to specific longitude and latitude locations. And then a lot of public databases capture environmental data that we can leverage and link with those locations. So the EPA has a lot of data about pollutants. The census data has a lot of information about social determinants of health, also the US Geologic Survey. And so I think that's a great way to capture the data that we need to pull into some of these analyses that we've been talking about. And in some ways, I think for some data types, it might be even more robust than asking people to share their information. So if you were to ask someone about their air pollution or water pollution, they probably don't know, but the EPA does based on where they live and where they work. And so that's one of the things that I've been thinking a lot about. The other, though, I think we could start to leverage participant reported information to tie into these analyses, either through surveys or also through wearables. We heard a little bit about this earlier today as well. And so I think the leveraging the phenotypic data that we can, and then trying to pull in all of these other data will really make the analyses a lot more powerful. Maybe before I finish, the one other thing I would say, Jim talked really briefly at the end of his talk about the PRS, and Nilajin might have more thoughts about this because I know this is a space he thinks a lot about too, but I want to echo something that Jim said in case somebody kind of looked away at that moment or wasn't paying attention. This idea of incorporating the environment with polygenic scores or polygenic risk scores, I think is really important as we think about future modeling. This isn't necessarily the multi-omics, this is really the genetics or maybe genetics with the expression. But where we incorporate those environmental variables is going to be crucial. So he showed us it's not necessarily sufficient to take a PRS term and the environment term in a linear model. The environment term may not be important in the context of the whole PRS added up together. It might be in subsets of the genes. We've also seen examples where we need to run the GWAS, incorporating the environmental variable in the GWAS to create the effect estimates in the summary statistic data set, and then incorporate it again when we build the polygenic score in the study data set that we're trying to estimate the score in. And so what we're seeing, we've been using BMI as a use case. I think it's a great kind of trait where we know environment is important, but for different environmental or social or behavioral variables, where you incorporate it into the model dramatically changes the performance of the polygenic score. And so I think this is early work that I think a lot of us are just starting to think about, but it's something that I think we need to be really thoughtful and intentional about where we put them and really think about the model that we're trying to build and the population that we're trying to estimate the risk in, or the polygenic score in. I don't think it's a one and done that we can just stick the variable in the model and it's going to work. So I might wrap up there and pitch it to someone else.
Fantastic. And before we go further with more questions, I would love to hear all of you, all the three panelists who joined us right now, about your general thoughts about the topic. I mean, we are talking about omics, multi-omics, and environmental exposures and cancer. I would like to hear from your experience. What are your thoughts also after hearing Jim and Christian giving their presentation? Maybe I'll start with Marina and then Nilanja, and then I'll go back to Marilyn.
Sure, sounds good. Hi, everybody. I'm Marina Sirota. Nice to meet you, and thank you for having me. I will say, in terms of my work, I've done quite a bit of multi-omics integration and also some environmental exposure work, but not in the context of cancer. So maybe I can share some of our learnings and then we can transfer that to potential cancer studies. We've done, a lot of my work is in the space of maternal health, and we've done a bit of work integrating environmental data using some of the approaches that Marilyn just mentioned based on geocoding with birth information to see which kind of exposures might be associated with adverse birth outcomes like preterm birth. In those types of analyses, what we've observed, for instance, using the Callan virus screen, which is a database, the California database, which captures zip code and census tract level information. But of course, there are challenges with that. That data is population level and not individual level. People move away. But nonetheless, you can use it to just get a proxy of what environmental exposures might be, and also capture some information from social determinants of health. We can use these types of data to drive hypotheses, which then can be followed up. So in our work, for instance, we identified that arsenic and nitrate as water contaminants might be associated with preterm birth. But then we can use other tools like mass spec and non-targeted approaches in specific samples and cohorts that we ourselves collect to follow up some of these hypotheses and associations. So that's, I guess, more in the context of just capturing environmental exposures. On the multi-omics side, one of the studies that we've led is a lupus study on a CLUES cohort, and it's a multi-ethnic cohort also in the state of California, where we have methylation genotyping, RNA-seq, as well as a non-targeted analysis of environmental exposures on the same cohort. And I guess in terms of integration, that's one of the challenges that was brought up earlier. It's a limited size cohort. It's about 350 individuals, but it's very deep phenotyping, both on the clinical side as well as on the molecular side on these patients. And we've been using different approaches, looking at clustering of the patients by their clinical data, and then associating with various omics as well as environmental factors. But it's actually a case-only cohort. It doesn't have any controls. And that is a huge challenge in a potential study design. So overall, the data is very deep, but that's another consideration. How do you match the cases and the controls, and whether you're able to do such deep phenotyping on a significant number of both healthy individuals and diseased individuals? And then finally, I'll touch upon leveraging publicly available data to answer some of these questions and integrate. And that's what quite a bit of work that we've been doing in the lab just generally. And there, the biggest challenge is you might have different types of measures from the public domain, but they might not be collected on the same patients. So there might be all lupus patients or all cancer patients, but they are different cohorts with different phenotypic heterogeneities. And we can use some of the phenotypes to bring these data together and start asking questions, but it's another kind of consideration to think about in terms of integration, and we can dive into specific examples. Of course, in the context of cancer, TCGA is fantastic, which captures the molecular side, but not much on the environmental side there that we can leverage. So I wanted to throw out some of these things and issues that we think about as we integrate both data on the molecular side, but also start bringing in some of the phenotypic and environmental determinants as well. So I'll stop there.
Thank you so much, Marina. I'll turn it to Nilanjan to share with us his experience.
Yeah, so that was fantastic, and thanks for inviting me to be a panelist here. And so I thought, I'll first talk about an issue that has been already raised. Jim very nicely presented an example, like how partitioning the PRS into pathway-specific effect, can better elucidate the gene-environment interaction. And I think to partition the PRS into pathways, I think there could be a big role of multi-omics analysis, where we have tried recently with the transcriptomic data, and then we're now trying with the proteomic data. I'm going to share in the chat, I'll share it later, a paper where he basically looked into the kind of GWAS-associated trait variants and asked the question that, and then we looked into large-scale transcriptomic data like EQTL Gen Consortium and asked the question that, can we look at it for a given trait, like a particular breast cancer, say, can we find combination of SNPs into that? We have currently about 200 or so breast cancer SNPs. Can we find combination of SNPs which can be associated with combinations of transcripts, and that the associations could be due to cis association, or it could be most of the time could be due to trans association. And then we so you can do this kind of like analysis through something called canonical correlation analysis, and there are a lot of packages for something called you can do sparse canonical correlation analysis. But what we found was interesting is that if you just do just look at the trans association between combinations of SNPs and combination of transcripts, so you have done also for proteins, you do find lots and lots of association, which are not disease relevant, because there is a lot of background association between genetic variants and proteome and transcriptome. But when we did propose a hypothesis testing framework where we said, okay, the kind of nature of association we see, is it more enriched compared to, for example, if we replace the set of SNPs, let's say breast cancer SNP, by a random set of SNPs in the genome, that could be associated with other traits and things like that. And that's when we see that kind of enrichment, and then we saw interesting patterns of relationship between the SNPs and downstream effect through the transcripts and proteome in a very cancer-specific way, disease-specific way are coming out. So I feel excited about that avenue, to kind of use transcriptomic, proteomic data and to establish how the PRS effects might be mediated through different downstream effects and identify those pathways. And then and then hopefully that will allow you to partition the PRS effects into different pathways. So we found some good experience in that. We first tried with the transcriptomic data. That's where our current paper is about, and now we are doing a paper on using the proteomic data and then doing a lot of very detailed follow-up analysis. And I think there is definitely we see some ability to kind of partition the PRS into distinct pathways. And then I thought another thing I will bring it up, which I haven't seen much discussion so far, is the role of the Mendelian randomization analysis and in multi-omics setting. And one idea I find it quite fascinating, and I haven't seen that much methodological work, and there is more need for that, is when you have multiple biomarkers for example, I'm interested, let's say, looking at whether inflammation causes risk of lung cancer, right? And then there could be specific biomarker like CRP, and we can do a Mendelian randomization analysis of CRP on lung cancer. A lot of the times you will not see a association if you do a Mendelian randomization analysis on CRP and lung cancer. And if you think about it and what's going on, CRP has, you have identifying genetic variants which predict CRP, and some of the variants might be affecting CRP through inflammation, but some of them are just biomarker specific variants, right? So so that's why we are not seeing the causal association. But if you have multiple biomarkers measuring the same inflammation, and which is kind of the same latent exposure, there might be better hope to actually try to test for the causal effect, because then we can look for instruments which are affecting multiple biomarkers at the same time, and those are more likely to be instrument, right, which are through the inflammation versus being a biomarker specific. And so I feel like the kind of the multi-omic may have a role in that way also, to kind of test for causal effects in the MR framework, but for not looking at individual biomarker, but for testing for the causal effects, something underlying latent variable like inflammation, and there are really good other examples. In that, I'll finish it there, and I'll post some papers on this.
Thank you so much. We love to see the papers as well. And I'll go back to Marilyn, please.
Well, I guess there's one more thing. I shared some of my earlier thoughts even in response to that first question anyway, but I want to go back to something that Marina said, that it's something that I think we all need to really start to think about strategies. And it's a really hard problem, but a really important one. And that is the sharing of public data. And this is, at the moment, I'm thinking more so on the multi-omics side than the environmental side. Only the environmental side, I think there's not nearly as much public data out yet, because we're just trying to figure out how to capture it well. But on the multi-omics data, there's a lot of data in the public domain. And I think with the new NIH data management sharing plan policies, we're only going to see the amount of multi-omics data in the public domain be higher and more. And one of the challenges that we face is that each data set on their own has a pretty small sample size, and they may have, 2 omics captured or 3 omics. And then another data set, even if it's of the same phenotype, they have 2 different omics. Or even if it's the same type, they both captured gene expression, but they did it using 2 different assays. Trying to think about ways that we can map the raw variables to some sort of standard representation of those biological phenomenon that we're measuring. So whether that's at the gene level or even at the pathway level, some way that allows us to start to think about integrating across the data sets, because no one's ever going to have a sample size, have the funding to generate a data set of a sample size of 1 million people with 5 omics assays, all done, in the same batches at the same time points, like all of the things we would like in the perfect design, we're just not going to have. But there's so much data out there that if we could figure out how to leverage it and bring other data sets to integrate with our data in sophisticated and biologically meaningful ways, I think maybe then we might gain some power in these multi-omic strategies. And like I said, I don't have the solution, but it is something I think spend a lot of time thinking about. And maybe it's partnering more with our molecular and cell biology colleagues that kind of, create and run these assays to think about, how do we take these raw variables and bring them together with the same types of raw variables in a different assay, but they're measuring really, the underlying biological phenomenon is the same thing, but how do we bring them together into models that allow us to combine data sets? And this assumes, of course, that we can deal with some of the study design differences across 2 studies. And we heard some about that in the previous session. And those are very important issues. So I don't want to suggest that we shouldn't also think about those as we do this, but if we had 2 studies that were designed in a similar way, but with different assays, how can we bring them together?
Fantastic. That is very helpful. And I would like to remind everyone that you have the option, everyone in attendance, that you have the option to ask questions and suggest questions in the Q&A box. So please do so. We have a couple already. But before going to the 2 on the Q&A, which are about EHR, I would like to ask another general question, which is, what are the new methods and approaches, particularly computational approaches, that we need to move the field forward, particularly when we want to integrate environmental data with existing other omics? And what do we expect with this? What will success look like in a way? What type of novel discoveries and how this will inform biological mechanisms? And I have 5 amazing colleagues here, and I look forward to anyone who wants to volunteer to kick us off with that. Elijah, please.
Yeah, so I mean, I thought Marilyn raised a very important question about the kind of related to data integration when you have different studies which are, and you can think in a more general question, the different studies have different strength, and some study have more focus on biomarkers, some study has more focus on exposures, and a very interesting methodological question is like, how do you develop model in terms of that can integrate both type of data, and what addition, for example, predicting a cancer risk model, and I think there's some good methodological work has been done, but I think we can learn from what has been done, for example, with the GWAS field. For example, people have shown that with the summary statistics data, you can have people might have model analysis done association for individual SNP1 at a time, and with a little bit of data on reference panel that allows you to estimate the correlation between the variables, the SNPs, you can try to fit a joint model, and that will include both SNPs. And similar principle can be applied for data integration in other contexts, where you might have a study which are providing information more on a particular types of exposures. There might be another study on which is giving more information on detailed information on another type of exposures. But what you need a little bit of data, not a lot of data, but you need a little bit of data to estimate the correlation between those variables. So you have to have this kind of reference panel, which are kind of it could be shallow data in the sense that you do not need a very, very large sample size, but you need to have a very deeply measured exposure data so that you can characterize the correlation between all types of exposure. And then you can use that information to integrate the other type of data, that the study data from the, so you can think about an equivalent of the 1000 genome, right? That gives a reference panel to estimate correlation between SNPs. And so we need that kind of data to which is, again, maybe small sample size, but very deeply phenotyped, and so that we can try to understand the correlation, and that information can be then transported into study data set which may be focusing on particular type of exposure, and then integrate that to build joint models.
Fantastic, thank you. Any reaction? And Christian, please.
This is actually great. So along the same vein, one of my interests is actually taking what we learn in one omics and seeing what can we learn about it in different omics. I mentioned, for example, that a frustration, it's a reality with this data. It's difficult when you get volunteers after disaster to provide every single data type that you want. On the other hand, so I'll give an example where we're doing a lot of microbiome profiling, and people have been put in databases where in the same individuals you have microbiome and metabolomics. With this kind of approach, you can take a microbiome signature and doing simple correlation metrics, figure out what are the matching metabolomic signatures. So realistically, one of my interests is to take, because again, we can only learn that much in real life with that we have, but I'm trying to take what we learn and apply it in whole different areas. So for example, a system like TCGA, where you can just focus on the normals, or databases where people do focus efforts to build references and measure the same, even in controls, measure the same omics, in different omics in the same individuals. This kind of approaches may allow us to bridge from one omic domain to a different omic domain. And some of them are more actionable. So if you give me gene expression, well, we can look for irreplaceable drugs and what have you. But literally having this kind of bridges, this would be, not ideally you have to measure everything everywhere, but practically having bridges like this would allow you to span more than your original discovery omics, So yeah, that's something I'm keen on.
So I totally agree with all that. And I think from a purely statistical standpoint, the ultimate power to detect an association between some exposure and some outcome is going to depend on all these various levels of correlation through the whole system. And every time you have a correlation less than 1, you're chipping away some of the power that you're going to be able to detect the real signal. So I think in thinking about any particular study where you want to put your resources, I think it's where you see the weakest links, where you see the weakest models for trying to integrate one omic with another omic. Because just having a poor model, in that system, if you're trying to link one to the other is really going to take a toll on the ultimate power that you have.
Fantastic. Anyone else has thoughts on this issue, or shall we move to the next questions and we can take those from the chat if that is okay? So in the Q&A, there are 2 questions that I think we can tackle at the same time about using electronic health records. And the first question is about when you use data on addresses, how do we deal with the past locations? So I guess when the address has changed, do you think the previous addresses or locations should be included in the analysis? Or maybe even we should account for the length of time that an individual has been at their current address. And the second question is also related to this, so I'll probably bring them up together, which is I'm a geospatial information scientist and I study neighborhood social determinants of health and environmental exposures. I'm very happy I can read the acronyms. Acronyms are daunting to me. And patient outcomes using electronic health records. What specific action can I take or study could I do to help? So I'll propose these 2 questions together, and would like to hear who wants to start first. Marilyn, please.
Sure. I'd love to collaborate with whomever you are, anonymous attendee, who does that type of work for a living. That'd be great, because we've done the geocoding and some of the linkages, and now we're trying to figure out how to use it, but that is not our expertise. So please feel free to reach out. But in all honesty, I do think it's an important piece, is connecting with the informaticians who are working closely with the EHR data, but they are not epidemiologists or social determinant of health experts, and also with then, the disease experts. So if it is somebody working in the cancer space or somebody, working in the women's health space, I think those partnerships and collaborations are really important. We all can't be the experts in everything. But the other point, I do think the past addresses and length of time is really important. And if you can capture the information, not only the length of time, but the period of time, so that then when you do the linkages, you can see kind of that window of time linked with a window of time in one of these public databases. Because as you can imagine, air pollution in a region is not static. It changes from month to month and year to year, as highways are under construction. Sometimes there's a region that's farmland that has very low air pollution, and then they build a highway that goes straight through that farmland area. And so now, 10 years later, the air pollution in that region is much higher. And so I think you do want to capture, if possible, not only the length of time, but actually the time period, so that when you do those linkages, you can try to match up their location with that time period in the publicly captured data.
Fantastic, Marina.
Yeah, I was going to comment both on the outcomes question and also the comment in the general chat about linking to to registry data potentially. And we do some HR data analysis too, again, not in the context of cancer, mostly in the context of women's health. But I will say, in my opinion, one of the biggest challenges with doing analysis relating to HR, whether it's looking at associations or predictive modeling or unsupervised learning and clustering, is outcomes, and being able to link and understand the relevant outcomes for a certain disease. And as I mentioned, in the context of women's health, one thing we look at is pregnancy outcomes, and that, nothing is really simple, but at least we have the deliveries in the hospital and we have some information about birth timing or what might have happened. At UCSF, we actually have a clinician curated separate database that curates every single delivery since 1970, which provides an amazing opportunity to mine those data, and it's somewhat structured. And for other conditions, there are different outcomes in the EHR, like lab test results, for instance, high cholesterol levels or high blood sugar levels. But for instance, for cancer patients, I don't even know. How do you know the outcome of a certain patient? And that's why, of course, linking to registry, I think, is very important, but also figuring out how to capture those outcomes, whether you look at clinical associations, or if some people have genetic data captured in their EHR, that's great. That's not true for us. But for cancer patients, I think there probably are ways to do that and link with some of the genetic testing that might have been done. I don't know. But also environmental exposures. How do you link that? How do you understand what the outcome measures are? I think it's a big question, and I don't have a good answer, but I thought I would bring it up.
Thank you. Christian, please go ahead.
Yeah, so just to mention that, these are the kind of questions that we thought in the past, or at least we provide, some sketches of how they address. So I really like the life history, question, because I could imagine, if you could do it at like a 6 months intervals, where was somebody, what was the air at the time, then, if we have this information, we have ways to deal with it, and like model with it. I just want to bring up that I think one of the issues that, so reality, the larger the input data sets and the higher quality, the better what you learn from them. So I really think, it's an issue of scaling. And frankly, I don't think it's something where scaling on the computational and it's something, we can deal with that. I think scaling the input and the quality of data is actually the biggest problem here. So I'm almost thinking, I don't know what kind of efforts would be required to try to compile such data as like databases that we can mine. Does it have to be state level? Does it have to be like national level? Because really, if all of us do studies in a few hundred, participants, we can learn something. But if we could do studies in tens of thousands, or bigger, that's where the real power would come. So I think ultimately one of the topics is how can you support scaling? How can you build input databases of high enough quality and large enough size? Because then I think we all can have methods that could deal with that, So yeah, I think, and that's a question also for the organizers, there's efforts to support that and the logistics for that. Yeah.
Fantastic. Any other takers or thoughts in response to these two questions? And again, there is, as mentioned, there is a paper that has been posted in the chat, so you may want to check it out. If not, I would like to propose a new question, which is especially going back, I think it's related to the question about the social determinants of health, especially what type of statistical tools and what type of approaches we need and we might want, or the field wants to develop or adapt to study multi-level and multidimensional structural and social determinants of health when we're doing multi-omics. And anyone wants to get us started?
I mean, the first thing we need is the more data on multi-ancestry population. As we know, like in the GWAS was very biased, and that was and that led to a lot of bias in the PRS analysis. And now, because of awareness, we are collecting more data, GWAS sample in multi-ancestry population. But there is similar bias in omics data. That a lot of omics studies that you hear a lot about, in the public domain, and they are done has been mostly in the European ancestry populations, and so there are downstream bias for that, because of that initial sample size issue. So the first thing we need, I would say, to understand the kind of how social determinants of health relates to the multi-omics aspect is that more data from multi-ancestry population calculating for the multi-omic data. And also I would add that also, unlike genetic variants, which is remains constant over time, the omics, of course, captures exposure, and we need to capture different exposure window. So we need to think about the not only collecting the multi-omic data, but we also have to think about collecting different exposure windows, starting from the pregnancy to the later in the life. And as a cancer, we think about it's a late onset disease, so you need to focus on the exposure window in the middle age or things like that. But there could be wonderful opportunities if we have the multi-omics data in multi-ancestry setting in early stage of life to establish the role of early life exposure on cancer risk. And I think we don't have much data on those things.
Thank you.
I can talk a little bit about maybe some of the analytical challenges. And one of them is figuring out how to capture longitudinal data. And I think we don't really know how to do that well across, even for a single type of measurement, it's not simple, especially not simple if we go back to the whole EHR discussion. But given multi-omic nature and trying to integrate different measures, where if you're lucky, you could have the measures taken at the same time per patient, but it could be different. So I think on the methodology side, again, I don't have an answer, but I have a question. I think a lot of development needs to happen in the next several years in that space.
And see, Marilyn was, and then Christian.
Yeah, I think I want to build on what Marina just said, and it's two parts. So we don't have a lot of longitudinal data, certainly for multi-omics, and I think starting to generate some multi-omics data on the same people at 3 different time points from the same tissue type would be incredibly useful to start to get a handle on what is the level of correlation over time. Do we see huge variability in the same people at 3 time points, maybe a couple years apart, or is it fairly constant? Or the expectation, I think, is that for some of the omics data types, they'll be fairly constant. There are probably others that are widely variable, and it's not even just the whole data type. Certain variables within that, certain genes are going to show huge variability in expression, and others are going to be fairly constant in, over the lifespan. If we're looking at development, like birth and middle age and old age, maybe there we see the differences, but we don't even have a lot of data to work with. I think we're mostly working on model systems and hypotheses at this point. But then once we have those data, if we do find that we need it over time, we also need better longitudinal statistical methods. So in the EHR data space, we talk a lot about how we have longitudinal data. We have 15 or 20 years of clinical data on someone, and yet for most things, we take the median LDL, the median BMI. We don't actually model it over time. There are statistical tools for kind of time series modeling, but most of those are done in not biomedical data types. And so they don't necessarily haven't been kind of adapted and applied in this space. So I think that would also be really useful. Maybe the one other thing I'd say, especially when we get to social determinants of health, the local ancestry methods have started to emerge, but they are still pretty limited in scaling. I think there's a few very new ones that are starting to look powerful, but as we think about the correlation between the social determinants and then the linkage with the omics data type, looking at, regional kind of ancestry and local ancestry is going to be important in the context, I think, of some of those variables.
Very important. I want to add that, especially with social determinants of health, sometimes there are traumas that are very limited in time, but incredibly powerful. And if we take averages over time, we're probably going to lose that. It's particularly important to think that way. And I think it was Chris and then Milan.
So obviously I agree with my colleagues. One other determinant I want to bring up, which is often, is not well developed, is actually diet. I mean, we especially, if we study, microbiome, but even metabolomics, often the lack of attention to diet. I mean, diet is a huge confounder. Okay, so I think it would be helpful at least to have, in some of the studies, make sure that we have a provision for that. I mean, again, diet is much more variable than, ancestry or location, but I think, at least having some control studies where we capture that would be important, at least, for certain data types. So if you look at, blood metabolomics, or like microbiome in the gut, definitely diet is a big factor. And again, diet cuts a bit across like, neighborhoods and ethnicities, also cultural aspect, but I think as a confounder is one that is often, it's not paid enough attention to.
Fantastic. Last word to Nilanja on this topic, and then we'll move to another one.
Yeah, just a comment on the data availability of the longitudinal omic data, that there is not that much data, but one study I do know of has valuable data is the atherosclerosis risk in community cohort, which is a cohort a lot used in the heart disease context. So it's not a large cohort in the sense like it's not a UK Biobank size cohort, but it is about 70,000 people. But they have very long-term follow-up, and those people have been visited 5 times, and they have collected biological sample, and now they have epigenome, proteome, and they're going towards transcriptome, metabolome on multiple visits. And so that's one study I know of. And that's also available in 2 ancestry, like European and African ancestry, but we definitely need more studies like that.
Thank you. We only have less than 5 minutes, but I have a question that's particularly dear to me. We have done some work in my lab about using DNA methylation to estimate exposures. Some of you might be familiar with epigenetic age. We can use DNA methylation to estimate someone's age. And some of us have been trying to measure risk factors, including environmental risk factors, using DNA methylation. So I think I would like to start from that to pitch a question about to what extent genomics, genetics, or omics data can be used to advance and impute exposures or identify latent exposure factors in the analysis of cancer studies. And Nilanja, you have your hands up, but it may be from before. If anyone wants to take this.
Before, I would.
Okay. But if you want to start now, you're also welcome.
I'll start. The obvious one comes from the kind of the example, like the example Jim was talking about. Like trying to build a PRS model to impute, biomarkers or other exposures, and that's a good place to start. But I still think that we should not just look for association. I think we should really think about causality here, and that's why I think instead of just building the PRS, I would try to preserve the individual SNP associations and then try to kind of disentangle what is coming from the horizontal pleiotropy versus the actual causal pathway, and really try to kind of establish a more of a causal link. I think we can, if we keep chasing association, and with a large volume of data with so many variables, we'll keep chasing a lot of things. I think we really need to start thinking in terms of more causal frameworks and how we can leverage the Mendelian randomization and in genetic instruments or other instruments to kind of more bringing in causal thinking into this kind of analysis.
Fantastic. Any other thoughts, and Marilyn?
Yeah, I wonder if it's possible to, in this examples where we do have the omic data and the exposure captured, to use, a data-driven approach to try to find, what are the, for example, the methylation patterns that are tightly linked with some exposure, such that then we could use that model to then impute that exposure in other individuals. And actually, in some ways, if we could figure out that link, that smoking causes these methylation marks in these genes, for example, then that actually would be a much more robust strategy to capture accurately. If that linkage is tight, that it actually is, smoking is causing these methylation marks to occur. It's not just an association, but it's causal. Then we don't have to ask people if they smoke or how much they smoke and hope that they're not lying. We could actually run the methylation assay and have that information captured. And so I think starting now to take a data-driven strategy to analyze the data where we do have both, identify those patterns, then I think it's a great strategy to think about imputing the ones that we don't have it captured.
And I would definitely loop SNP genotypes in there as well into that modeling. Anything you've got reliably on, big cohorts, you want to feed that into your submodels to try to use as much as you can.
And I see Catherine has lit up, so I'll leave it to her to close the session. But I would like to say that, Marilyn, your example is great because with DNA methylation, actually, there are predictors that are data-driven to estimate smoking, that can estimate lifetime smoking, that seem to work better than self-reported smoking in most cases. So that's something that's interesting to think about. Catherine, it seems you have a contribution to give us, so I'll leave it to you. Unmute yourself.
Sorry, I was going to, I actually agree with what Marilyn has said, that you really need to have that tight link between the exposure and the methylation patterns that you see, whether you're talking about methylation or some other epigenetic mark. But once you do that, I have a feeling that because this is going to differ, say, by age or by the timing at which this the DNA you are using was collected relative to exposure, it's probably worthwhile to think about doing this multiple times cross-sectionally over time. And I very much agree. I think that's if we could do that, I think we will loosen up a lot of things that we are having difficulty. Yeah, that's all I wanted to say.
Fantastic. And there are a few comments on the chat that probably we don't have time to go to. And the Q&A, I think there is one resource on NCBI PBIT that about NCI data ecosystem you might want to check out, and you'll see it there. If Leah or Kim have more information on this, please let us know. But I think this brings this session to an end. I want to thank everyone, the two speakers and all the panelists. This has been amazing and very informative. And we look forward to the open session, and I'll hand off the screen to Kim at this point.